In this video today we are going to read about one of the metabolism of carbohydrate, that is glycolysis. Before starting the metabolism glycolysis, I would like to go in brief about the history of the glycolysis. The glycolysis is derived from the Greek words glycose, that is sweet or sugar, or lysis means that is dissolution. It is a universal pathway in the living cells. The complete pathway of glycolysis was elucidated in 1940. This pathway is often referred to as Eden-Meyerhof pathway (EM pathway) in the honor of the two biochemists who made a major contribution to the knowledge of glycolysis. What is glycolysis then? I will start with the short definition. The glycolysis is defined as the sequence of reaction converting glucose or glycogen to pyruvate or lactate with the production of the ATP. Previously, I have already described this in the various terminologies of the carbohydrate metabolism, is that the glycolysis is the oxidation of glucose to pyruvate and lactate with the production of the ATP. Now let us discuss about silent features of the glycolysis before actually going towards the topic of glycolysis. The silent features of glycolysis are: the glycolysis takes place in all cells of the body. The enzymes of this pathway are present in the cytosomal fraction of the cell. The glycolysis occurs in the absence of oxygen, that is anaerobic condition, or the glycolysis can also occur in the presence of oxygen, that is the aerobic condition. You have to know that the lactate is the end product under the anaerobic condition. In the aerobic condition, the pyruvate is formed and later on the pyruvate is oxidized to carbon dioxide and water. The glycolysis is also a major pathway for ATP synthesis in the tissue that is lacking mitochondria. The tissues that are lacking mitochondria are erythrocytes, cornea and lens. Those are the tissues that are lacking mitochondria, and glycolysis is the major pathway for the ATP synthesis in those tissues. You have to know that better. Glycolysis is very essential for brain, which is dependent on the glucose for energy. The glucose in the brain has to undergo glycolysis before it is oxidized to carbon dioxide and water. The glycolysis under anaerobic condition may be summarized by the net reaction, that is glucose plus 2 ADP plus 2 phosphate ion, which gives 2 lactate plus 2 ATP. This anaerobic glycolysis reaction, net reaction, you should know very well because there is production of the 2 lactate and 2 ATP under the anaerobic condition. The glycolysis is a central metabolic pathway with many of its intermediate providing branch point to other pathway. Thus, the intermediates of the glycolysis are useful for synthesis of amino acids and fat. This is the very important thing that you should know. The reversal of glycolysis along with the alternative arrangement at the irreversible steps that will result in the synthesis of the glucose, that is known as gluconeogenesis. The gluconeogenesis, I will discuss that detail in another video, another lecture. Now, after knowing the silent features of the glycolysis, I want you to know the reaction of the glycolysis. The sequence of reaction of the glycolysis is I have given shown in this picture. You can see in this picture, this is the sequence of reaction of glycolysis, and the pathway can be divided into 3 distinct phases. One is energy investment phase or priming stage. The another is splitting phase. The another phase is energy generation phase. These are the 3 phases of the reaction of the glycolysis. Again, I am going to say is one is energy investment phase, another splitting phase, and last one is energy generation phase. After knowing 3 distinct phases of the reaction of the glycolysis, I want to start the glycolysis by the energy investment phase. Now let us move towards the first reaction of the energy investment phase, that is conversion of the glucose to glucose 6-phosphate. You have to know in this first reaction is that glucose is phosphorylate to glucose 6-phosphate by hexokinase or glucokinase. Both the hexokinase and glucokinases are the isoenzyme. This hexokinase and glucokinase, hexokinase versus glucokinase topic I have already discussed in the previous video. I want you to see that. So this glucose phosphorylation to glucose 6-phosphate is an irreversible reaction which is dependent on the ATP and magnesium. The enzyme hexokinase is present in almost all the tissues. It catalyzes the phosphorylation of the various hexoses like fructose and mannose. The hexokinase has low Michel's-Menten constant and is inhibited by the glucose 6-phosphate. Whereas the glucokinase is present in the liver, it catalyzes the phosphorylation of only glucose. It has high Michel's-Menten constant for the glucose and is not inhibited by the glucose 6-phosphate. You have to know that. Due to the high affinity of low Michel's-Menten constant, glucose is utilized by the hexokinase even at the low concentration, whereas the glucokinase acts only at the higher level of the glucose. That means for after meal, when the glucose concentration is above 100 mg per deciliter, the glucokinase acts to store the excess glucose to the glycogen in the liver. You have to know that glucose 6-phosphate is impermeable to the cell membrane. The glucose 6-phosphate is a central molecule with a variety of metabolic fates. The glucose 6-phosphate can go to the glycolysis metabolism, it can also go to the glycogenesis, or it can also go to the gluconeogenesis, or it can also go to the pentose phosphate pathway. You have to know this very well. Now in another reaction of the energy investment phase, the glucose 6-phosphate undergoes isomerization to give the fructose 6-phosphate in the presence of the enzyme phosphohexose, isomerase and magnesium. Now let us move towards the last reaction of the energy investment phase. Here the fructose 6-phosphate is phosphorylated to fructose 1,6 bisphosphate by phosphofructokinase. This is an irreversible and regulatory step in the glycolysis. So these are the three reaction of the energy investment phase that you should know. Now let us move towards the another phase of the glycolysis, that is splitting phase. The splitting phase you have to know is that the 6-carbon fructose 1,6 bisphosphate is splitted, hence the name glycolysis, to 2 3-carbon compounds. One compound is glyceraldehyde 3-phosphate and another compound is dihydroxyacetone phosphate by the enzyme aldolase. The enzyme phosphotriose isomerase catalyzes the reversible interconversion of the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this is the very crucial step. Here the 2 molecules of glyceraldehyde 3-phosphate are obtained from 1 molecule of glucose. So this is the very important step that you should know in the splitting phase. Now let us move towards another phase, that is energy generation phase. In the energy generation phase, what happens? The glyceraldehyde 3-phosphate dehydrogenase converts the glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate. This is an important step. It is involved in the formation of NADH plus H plus and a high energy compound 1,3 bisphosphoglycerate is produced. The lodoacetate and arsenate inhibit the enzyme glyceraldehyde 3-phosphate dehydrogenase. In aerobic condition, the NADH passes through the electron transport chain and 6 ATP are synthesized by the oxidative phosphorylation. That you should know this very well. Now let us move towards the another step of the energy generation phase, that is the enzyme phosphoglycerate kinase acts on 1,3 bisphosphoglycerate, resulting in the synthesis of the ATP and formation of the 3-phosphoglycerate. You should know that this step is the good example of the phosphate substrate level phosphorylation. It is called substrate level phosphorylation because the ATP is synthesized from the substrate without involvement of the electron transport chain. The phosphoglycerate kinase reaction is reversible reaction and it is a rare example among the kinase reaction. So this question may also be asked in the MCQs. What is the reversible reaction of the kinase reaction? That is phosphoglycerate kinase reaction in the glycolysis is the reversible reaction, which is the rare example. And then after that, what happened? The 3-phosphoglycerate is converted to the 2-phosphoglycerate by the phosphoglycerate mutase. This is the example of the isomerization reaction. The high energy compound phosphoenol pyruvate is generated from the 2-phosphoglycerate by the enzyme enolase. This enzyme, the enolase, requires magnesium or manganese and is inhibited by the fluoride. For the blood glucose estimation in the laboratory, fluoride is added to the blood to prevent glycolysis by the cells. So that the blood glucose is correctly estimated. The fluoride combines with the magnesium and phosphate to form a complex that binds with active site of enolase and blocks access of the substrate. Thus, the fluoride is an unusual competitive inhibitor. This is one of the application of the fluoride inhibition in the glycolysis that you should know very well while estimating the blood glucose. The enzyme pyruvate kinase catalyzes the transfer of high energy phosphate from phosphoenol pyruvate to ADP, leading to the formation of the ATP. This step also is the substrate level phosphorylation and this reaction is irreversible. So finally there is formation of the pyruvate. Now let us discuss the conversion of pyruvate to lactate. What is the significance of conversion of pyruvate to lactate? You have to know that under anaerobic conditions, which is lack of oxygen, the pyruvate is reduced by the NADH to lactate in presence of the enzyme lactate dehydrogenase, which is the competitive inhibitor oxamate. The NADH utilized in this step is obtained from the reaction catalyzed by the glyceraldehyde 3-phosphate dehydrogenase. The formation of the lactate allows the generation of the NAD+, which can be reused by the glyceraldehyde 3-phosphate dehydrogenase so that glycolysis proceeds even in the absence of the oxygen to supply the ATP. So main significance of the conversion of pyruvate to lactate is that the glycolysis to proceed even in the absence of the oxygen to continuous supply of the ATP. The occurrence of the uninterrupted glycolysis is very essential in the skeletal muscle during strenuous exercise where the oxygen supply is very limited. The glycolysis in the erythrocyte leads to the lactate production because mitochondria, the centers of the aerobic oxidation, are absent. Brain, retina, skin, renal medulla, gastrointestinal tract derive most of their energy from the glycolysis. This you should know very well. So now we have completed the 3 phases of the glycolysis: energy investment phase, splitting phase and energy generation phase. And we have read all the reaction in the energy investment phase, splitting phase and energy generation phase. And also I have described you the each and every enzymes and describe you the ATP consumption or ATP production or NADH production. Now let us revise the whole glycolysis metabolism, whole pathway of the glycolysis. Let us revise. First of all, the glucose. The glucose by the action of the hexokinase or glucokinase is converted to glucose 6-phosphate with the consumption of the ATP. So the glucose 6-phosphate is again metabolized to fructose 6-phosphate by the enzyme phosphohexose isomerase. So this is the main thing you have to know. After conversion of the glucose 6-phosphate to phosphohexose isomerase to fructose 6-phosphate, the fructose 6-phosphate is metabolized to fructose 1,6 bis-phosphate by the enzyme phosphofructokinase by the consumption of the ATP. The fructose 1,6 bis-phosphate is metabolized or changed into 2 compounds, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by the action of the enzyme aldolase. What happens is that the dihydroxyacetone phosphate is also interchangeable to glycerol D3-phosphate by the action of the phosphotriose isomerase. This is the crucial steps that you should know. Here generation of the two molecule of glycerol D3-phosphate is obtained. After obtaining the molecule glycerol D3-phosphate, the glycerol D3-phosphate is metabolized to 1,3 bis phosphoglycerate by the action of the enzyme glycerol D3-phosphate dehydrogenase. Here there is conversion of the NADH. I mean there is production of the NADH plus S plus. And what happened? 1,3 bis phosphoglycerate is metabolized to 3-phosphoglycerate by the action of the enzyme phosphoglycerate kinase with the production of the ATP. The 3-phosphoglycerate is converted to 2-phosphoglycerate by the action of the phosphoglycerate mutase. The 2-phosphoglycerate is converted to phosphoenol pyruvate by the action of the enzyme enolase. The phosphoenol pyruvate is converted to pyruvate by the action of the enzyme pyruvate kinase by the production of the ATP. So this is the substrate level reaction. So this is the substrate level phosphorylation that you should know. In the glycolysis there are two substrate level phosphorylation. First substrate level phosphorylation is conversion of the 1,3 bis phosphoglycerate to 3-phosphoglycerate where there is production of the ATP, and another substrate level phosphorylation is the conversion of the phosphoenol pyruvate to pyruvate by the enzyme pyruvate kinase. So this is the main reaction. At last, what happens? The pyruvate enol form is converted to keto form. There is spontaneous changes, and when there is anaerobic condition where there is lack of oxygen, the pyruvate is changed into lactate by the action of lactate dehydrogenase. So NADH is utilized in this step. So this is the main all the glycolysis steps, various steps and various phases of the glycolysis that you knew. I have described in this video. I hope you understood this lecture. Thank you.